Hello students, welcome to the 12th class of Immunology. In this class, we are going to discuss the B cell development pathway. So, before going ahead, let us see what we discussed in our last class. So, in last class, we discussed about the T cell development. So, we started with the thymus, then different T cell development stages, then different type of T cell maturation, and some tolerance mechanism. In continuity to our last discussion, now today we are going to start with the B cell development. Okay, so let's start. Now, if we see the B cells, so B cells develop in bone marrow. So, bone marrow is the site of the B cell development. All right, that's why the name B comes here. And a hemopoietic stem cell, it becomes common lymphoid progenitors, which is CLPs, and these CLPs can give rise to B cells, T cells, or innate lymphoid cells. Okay, so in our last class also, we saw how the CLPs, they determine or decide whether to go towards the T cell pathway or towards the B cell pathway, and it was basically the notch. Correct. Now, majority of the CLPs in the bone marrow, they enter the B cell pathway. That means most of the cells, they become the B cells. And these CLPs, which are the common lymphoid progenitors, they undergo a series of differentiation to form immature B cells. So here you can see these are the stem cells, and then they are forming the early pro B cells. So this is the pro B cell stage, which is divided into early pro B and pro B or pre B stage, and then that is followed by the immature B cells. So the pro B stage here, it is irreversibly committed to B cell lineage. So once the CLP, it enters into the pro B cell stage, then it is irreversibly committed to B cell lineage. It will not form any other kind of cell. The recombination of the Ig genes begin. So if you can see here, the DJH chain recombination, it starts in the early pro B cell stage, and then start of the VDJH, that means the heavy chain recombination, starts here. Okay, after completed cell surface IgM expression, the immature B cells, they move to spleen to complete its maturation through the transitional T1 and T2 stages. So if you can see here, from the early pro B, it forms the pro B or pre B, where we have the complete VDJ heavy chain recombination, we have the clonal expansion, and then we have the VJ light chain recombination completed here. From this stage, the next stage in the development comes is the immature B cells, where we have or we see the negative selection, where the deletion and receptor editing take place. And from here, it goes as to the spleen as a T1 cells, or we call it as a transitional 1 cells, and then T2 cells, they form the transitional 2 cells. So what are these T1 cells, T2 cells? We will see in the later slides. Now, if we see the overview of the B cell development, then this picture gives a complete, brief. Now, if you can see here, we have the hematopoietic stem cells. This cell is a hematopoietic stem cell. And if you can see here, these are the arteriolar blood vessels which have the endothelial cells. These pink color cells are the endothelial cells, and these green color cells, this is the CAR cell, which is CXC12 abundant reticular cell. Okay, this particular cell. And they also have the mesenchymal stromal progenitor cell, which is the MSPC, mesenchymal stromal progenitor cells, and they surround this arteriolar blood vessels. Now, this cell, which is CAR cell, it releases or secretes CXCL12, which is the chemokine ligand CXCL12, and SCF, all right, stem cell factor. And these cells, the mesenchymal stromal progenitor, they also secrete the CXCL12, right? The same which was secreted by these CAR. Now, once these cytokines they are released, they prime the hematopoietic stem cells and they form the lymphoid primed hematopoietic stem cells. All right. So now the hematopoietic stem cell is primed, okay, under the influence of CXCL12 and SCF. Now what happens? Now this lymphoid primed hematopoietic stem cell through the notch signaling with the endothelial cells as well as with the pre-osteoblast secreting IL-7 and osteoblast secreting CXCL12 and the SCF secreted by these CAR cells and IL-7, okay, they differentiate into CLP, which is the common lymphoid progenitor cell. So now this is common lymphoid progenitor cell. Okay. Now we have the pre-osteoblast here which secrete the CXCL12 and IL-7, right? Now they make these cells to differentiate into pre-pro B cell. So this is pre-pro B cell differentiation, all right? And the osteoblasts and mesenchymal stromal progenitor cells, they through the WNT signaling and the pre-osteoblast cells through IL-7 and the CAR cells and sinusoidal endothelial cells, they secrete IL-7 and they differentiate the cells, the pre-pro B cell into pro B cell. Now the cell becomes the pro B cell. Clear? Now what happens? The pre-osteoblast, they also secrete IL-7 and GF1, all right, and then the IL-7 secreted by the sinusoidal endothelial cells and adipocytes secrete the leptin and they differentiate the pro B cell into pre B cell. So now our cell is a pre B cell. Okay, then we have the stromal cells and through the galectin 1 binding they differentiate into pre-B cell to pre-B cell which are slightly more advanced towards the differentiation pathway, and then they form the immature B cell, and that's how the completion from the hematopoietic stem cell to immature B cell development, it take place inside the bone marrow. Okay, now if we see, the bone marrow has niches or specialized microenvironments. What's a niche? It's a specialized microenvironment for B cell development. So if you can see here, we have the bone, this is the bone marrow cavity, and then we have this endostele region, then we have the sub endostele region, which is just under this endostele region, then we have the central region, and then we have perisinusoidal region. So these are some of the regions of the bone marrow cavity. So B cell development is regulated by spatially distinct BN niches. So that means, what do you mean by spatially distinct? That means they have these niches which are divided by space. Okay, so if you can see here, we are talking about the specific niches or specific microenvironments which are present at specific location inside the bone marrow. The current hematopoietic stem cell niche model, it consists of two distinct niches. One is the endostele niche and other is the central niche, which are distinct by their location and cellular composition. Simple. So that means these two niches, one is the endostele niche, so this is the endostele region, and then we have the central niche, So this is the central region from this to this. These two niches, they are important for this particular HSC niche model. Okay. Now, if we compare these two, endostele niche and central niche, then endostele niche is enriched with osteoblasts and osteoclasts. So basically, if you see this region, they are very heavily populated by osteoblasts and osteoclasts. And the central niche, which is this region, it is rich in perivascular MSPCs. So here you can see this is the perivascular region, and here this is the perivascular MSPCs. And now this endostele niche, they contain the transition zone blood vessels. So you will find the transition zone blood vessels in this endostele niche, while the central niche contains the perisinusoidal and periarterioral blood vessels. Okay, these two kind of blood vessels are present in central niche. The endostele niche has the transition zone blood vessels, which are surrounded by perivascular mesenchymal stem cell and progenitor cells. So these two kind of cells, they surround the endostele niche, whereas in case of central niche also surrounded by the perivascular MSPCs. Clear? So this is the comparison between the endostele and the central niche, which is our current understanding about the hematopoietic stem cell niche model for B cell development. Now, if we see the B cells appearance, so in case of mice, then the gestation period in mice is 19 to 21 days, and the hematopoiesis begins around 7 days post fertilization. Okay, so this you can see, this is the timeline here. This is 7.5, this is 10.5 days, 12.5 days, 15.5 days, and then this is the birth. So fetal hematopoietic stem cells in the early aorta-gonad mesonephros, which is AGM region, on D8. So if you can see here, this is somewhere around D8. All right, and this is the AGM region here. By D11, that is day 11, in the yolk sac, placenta, and fetal liver. So initially it starts in the AGM, but slowly by 11th day it moves to the fetal liver, to placenta and yolk sac. Okay, this is yolk sac, this is fetal liver, and this is placenta, and you can see they are somewhere by day 11. If I mark it here, then they are overlapping. These three are overlapping here. Now, in day 11.5 and 12.5, there is a fast expansion of placental hematopoietic stem cells. So you can see here, this is the fast expansion of hematopoietic stem cells in placenta. By day 13.5, which is day 13.5, placental hematopoietic stem cells, they begin to fall, but in liver continues to expand. So if you can see here, around this is 12.5, this is 15.5, somewhere here in 13.5, the placental hematopoietic stem cells, they are declining. Okay, so slowly they will stop here, but the fetal liver, they continue. All right, the expansion continues here. After complete embryo development, sorry, the main site of hematopoietic stem cell generation is the fetal liver, but some hematopoiesis in the spleen around birth. So you can see here that fetal liver is here, and they are the main site of HSC generation. But then some is also seen in spleen. Pre-B cells are first observed at day 13. So if you can see the pre-B cells, they are first observed at day 13, and hematopoietic stem cells, they first seed the bone marrow at approximately day 15. Okay. In few weeks, the bone marrow is the main site of B cell development for life. After few weeks, it is the bone marrow which becomes the main site of B cell development. Now, if we see the human, so here you can see, we have compared, this is the mouse timeline and this is the human timeline. Okay. In humans, it is the similar sequence but elongated time frame. The time frame is slightly elongated. So blood cell precursors first appear in the yolk sac in the third week of embryonic development, but not capable of generating all subsets of blood cells. The first cells to repopulate whole adult human hematopoietic system, they arise in the AGM region of the embryo and or the yolk sac. That means either or, either in this and or this plus this, in both the cases, in both the places. The first cells which repopulate the whole adult human hematopoietic system, they arise here in AGM. By third month of the pregnancy, hematopoietic stem cells, they migrate to the fetal liver. Okay, they migrate to fetal liver, and it is the major site of hematopoiesis in the fetus. So remember that the major site of hematopoiesis in humans during the fetus is liver. By fourth month, hematopoietic stem cells migrate to bone marrow, which slowly assumes the hematopoietic role until by the time of birth, and it is the primary generative organ for blood cells. And before puberty, most bones are hematopoietically active, but by the age of 18 years, only the vertebrae, ribs, sternum, skull, pelvis, and parts of humerus and femur. Rest of the bones, they stop, hematopoietically active. Okay. Now, if we see the later stages of B cell development, so the stages of B cell differentiation, they have two systems of nomenclature. Let us first see, what are the systems of nomenclature. The first is the basal nomenclature. It was developed by Malkers et al. And according to this nomenclature, the cells can be named as pre-B cells, pro-B cells, pre-pro B cells, pro-B cells, pre-B cells, and immature B cells. And it is the most widely used. In papers, you will find, this nomenclature, the naming will be very, very common. While the second nomenclature is the Hardy nomenclature, which was developed by Hardy et al. And according to this, the different stages, developmental stages, are assigned the letters like A, B, C, C prime, D, and E. So you can compare that the pre-pro B cells are A, the early pro is B, the late pro is C, and then this is the first checkpoint here in the late pro B cell. Then there is a large P cell which is C prime, then there is a small P cell which is D, and finally the immature B cells which is equivalent to E. And here the immature B cells, they also display the second checkpoint. So we have the two quality checkpoints at one at the late pro B cell stage and another at the immature B cell stage. So we will see. Okay. Now the expression of selected marker proteins, it is correlated with the extent of Ig gene rearrangement during B cell development from the pre-pro B cell to the immature T1 B cell stage. So basically, we see the marker protein expression, and then on the basis of these marker proteins, we determine what is the current stage of a particular B cell during the development cycle. Okay. Now, if we see the later stages of the B cell development, then this is the pre-pro stage where we have the germline. Okay, this is the germline rearrangement of heavy and light chain. While there is no surface Ig receptor expression, c-KIT is absent, IL-7R, which is receptor for IL-7, is very low, CD25 and CD19 absent, and B22, which is the CD45, this is present. In case of early pro B cell or the B cell, according to different nomenclature system, we have the DJ heavy chain. While the surface Ig receptor expression is absent, c-KIT has started in a low expression. IL-7R is either low or it is in good expression, and CD25 is still negative, CD19 is positive, and B22 or CD45 is positive. In the late pro B stage or the C stage, there is some V heavy D J heavy chain expression, and surface Ig receptor expression is none. c-KIT is low, IL-7R positive, CD25 is positive or negative, CD19 is positive, and B22 CD45R is positive. Now, if we see after completing the first checkpoint, it moves towards the large pre-B cell stage where the V heavy and DJH heavy chain, recombines, and then we have the first pre-BCR, which is the B cell receptor. While the c-KIT is absent, IL-7R receptor is present, it's positive, CD25 is positive, and CD19 is positive. And as usual, the B220 or CD45R is positive as well. Now it moves to the small pre-B cell, right? Here we have the V heavy and DJ heavy chain and V light and JL rearrangement begins here, and the surface IG receptor, it decreasing levels of pre-BCR, c-KIT is absent, IL-7R positive, CD25 positive, CD19 positive, and then B220 or CD45R is positive. And then comes the immature B cell, which again, we do the second checkpoint here, but in this particular cell, we have the V heavy D J heavy chain and V light and J light chain present there, and then we have the IgM expression, which happens here first time here in case of the immature B cell. So remember this, and then we have the c-KIT negative, IL-7R positive, CD25 positive, CD19 positive, and B20 CD45 are positive. Now if you see among all the markers, B220 or CD45 is present in each and every stage. All right, starting from the pre-pro to the immature B cell. And that's why this is a very reliable B cell lineage specific marker. Okay, B220 CD45R is a B cell lineage specific marker because it is present during all the developing stages in B lymphocytes. Correct? Okay. Now let's discuss about the checkpoints in the B cell development. So we have the pre-B cell checkpoint, which is the first checkpoint here we saw in our last slide. So the pre-B cell stage is tested, okay, to determine if they can express a functional BCR heavy chain in combination with variable pre-B and lambda 5 to form the pre-BCR. So that means the pre-BCR consists of a BCR heavy chain and variable pre-B and lambda 5. Okay. So V, pre-B and lambda 5 genes, they do not belong to the light chain gene families. Right? Remember, they do not belong to light chain gene families. V pre-B, it is homologous to a light chain V region domain with extra 25 amino acids at the C terminus. That is the difference. Okay, it has an extra 25 amino acids at the C terminus, while the lambda 5 is homologous to lambda light chain and J plus C sequences, which is J plus constant region sequences with extra 50 amino acids at the N terminus. So you remember in V pre-B, at the C terminus there are 25 extra amino acids, while in lambda 5 there are 50 amino acids at the N terminus. Now what is the function of these extra amino acids? These extra amino acids, they cover the heavy chain CDR3 to prevent it from recognizing any antigen. The main function is that they cover the heavy chain CDR3 to prevent it from recognizing any antigen. There are multiple pre-BCRs which self-aggregate. They initiate signaling with CD19 co-receptors to initiate further differentiation. Clear? Now mice need IL-7 receptor signal too, but humans don't need it. That means mice need IL-7 ligand to bind with receptor, and then apart from other signals, but we humans, we don't need it. B cells that fail to form a functional pre-BCR, they are eliminated by apoptosis, and that's why it is the checkpoint. All right. So B cells that fail to form a functional pre-BCR receptor, they are eliminated by apoptosis. Okay. Now, immature B cells also have the second checkpoint at this particular stage. So, immature B cells have only functional surface IgM. We saw the 2 slides back, and these are tested for the successful rearrangement of both heavy chain and light chain genes, and if they can express the IgM BCR. The cells with autoreactive receptors, they undergo one of the three fates. If they are determined by our system that the BCRs are autoreactive, so they will undergo three fates. What are three fates? First is that some are lost before leaving the bone marrow by apoptosis. So we call it as a clonal deletion. Correct. The second possibility is that others, they reactivate their RAG genes for light chain receptor editing. So they will be given a chance so that they can edit their receptors so that they not become the self reactive. And the third fate is that some may survive to escape the bone marrow but become anergic or unresponsive to any further antigenic stimuli. All right, although they will escape, and they will escape the bone marrow, but they will not be activated, they will be unresponsive to antigen. So these are the three fates which the autoreactive receptors during the immature B cell stage they can undergo. So elimination of self-reactive cells in the primary lymphoid organs is called central tolerance, whereas the elimination or becoming energetic at other places are called peripheral tolerance. So this is the difference between the central tolerance and peripheral tolerance. Now let us see the transitional B cells. Okay, you know that when they leave the bone marrow and then when they go towards spleen, then they becomes the transitional B cells. So immature B cells, they reaching to spleen from bone marrow, they are known as the transitional 1 or T1 B cells. And the interaction with self antigens in spleen, it can induce these cells to undergo apoptosis, and this is a part of the negative cell action. Okay. Now T1B cells, when they enter the follicles where IgD expression increases, it becomes the T2B cells, and these T2B cells, they populate the follicles of lymph nodes and spleen, and they become the follicular B cells. All right. So basically when T1B cells, they increase the IgD expression and they are called the T2B cells, and these T2 or the transitional 2B cells, when they populate the follicles of the lymph node and spleen, they call it as a follicular B cells. Now signals from the cytokine BAFF binding to the BAFF receptor, they are necessary for the survival of transitional and mature B cells. Without this signal, the cells will not be surviving. Okay. Some transitional cells, which we call it as a T3, okay, they are energized by exposure to weakly stimulating peripheral self antigens. So basically these T3 cells, they have the energy, they are unresponsive, they are energized by exposure to weakly stimulating peripheral self antigens, and these are the soluble proteins. Now these energetic B cells, they have a shorter half-life, and possibly they excluded from the B cell follicles in the lymph node and spleen, but more investigation is needed. Okay. Now let's talk about the mature B cells. So fully mature B cells, they express high levels of IgD and intermediate levels of IgM on their surface. So you remember that most of the proteins or BCRs, they have the IgT types, while they have the intermediate levels of IgM on the surface, although their specificity remains the same. There are three major populations of the mature B cells in the periphery. Three major populations. We have the follicular B cells, we call as B2 cells. Then we have the B1 B cells, and then we have the marginal zone B cells, or we call them as the MZ B cells. The follicular B cells niches, they have a limited capacity, and that once full, okay, this capacity is full in the follicle, they turn away additional B cells most probably due to competition for survival factors, particularly BAFF and its related proteins. So you saw in the last slide that BAFF is very important for the survival. Follicular B2B cells, which is the second type of cells, sorry, which is the B2B cells, they have a half-life of approximately 4.5 months. In contrast, since B1 B cells can self-renew in the periphery, their numbers are unaffected. Okay, so B2 B cells, they keep replaced by new cells as their half-life is approximately 4.5 months. Now, if we compare the follicular B2 B cells, B1 B cells, and marginal zone B cells, then the major sites in follicular B2 B cells are the secondary lymphoid organs, which are SLO. In B1 B cells, it is the peritoneal and pleural cavities, while marginal zone B cells, they are the marginal zones of spleen. Okay. Now, if we see the progenitors, they first appear in mice hematopoietic stem cells on day E10.5, while progenitors on day E9.5, and from hematopoietic stem cells also earlier progenitors. Okay, if we see the source of new B cells in adults, this is the hematopoietic stem cell in bone marrow. They are self-renewing, so division of existing B1 cells, and from again hematopoietic stem cells in bone marrow, and they are slightly long lived. So marginal zone B cells are long lived. Now, if we see the dependence on IL-7 and BAFF, then definitely yes. In case of follicular B cells, B1 cells, no, while marginal zone B cells, yes. If we see the V region diversity, the follicular cells are highly diverse. The B1 B cells are restricted. They show restricted diversity. They have a limited VH and VL uses and N nucleotide addition. Whereas in marginal zone B cells case, it is somewhat restricted. Now, if we see the phenomenon of somatic hypermutation, which we will discuss later, it is extensive in case of follicular B cells. It is some, event is seen in B1 B cells, while it is unclear whether the marginal zone B cells, they undergo somatic hypermutation or not. The requirements for T cell help is needed in case of B cells, follicular B cells, and not required in B1 B cells, while in marginal zone B cells it is variable. Some cells require, some cells don't require. If we see the isotypes which are produced by the follicular B cells, there are high levels of IgG. In case of B1 B cells, primarily they are IgM and some IgG, while in case of marginal zone B cell, they are primarily IgM and some IgG, as the B1 cells. If we see the response to carbohydrate antigen, in case of follicular B cells, it is possible. In case of B1 B cells, yes, and yes for marginal zone B cells as well. The response to protein antigen is definitely yes, and in B1 B cells it is possible, and in marginal zone B cells it is yes. Now, if we see the memory production, yes in case of B2 B cells, in case of B1 B cells, while in marginal zone B cells it is unknown. Now, this is the structure of, or the diagrams depicting the follicular B cells, B1 B cells, and marginal zone B cells. So here you can see different receptors here. Okay, and the markers. So you can see here in follicular B cells we have CD19, CD21. Here also in marginal zone B cells we have CD19, CD21, while it is not here. While we have follicular B cells CD25, which is absent in both, and here we have CD5 in the B1 B cells, and the marginal zone B cells we have CD1, right? And these are the IgD. It is low, IgD low, it is IgD high, and this is IgM, it is low, IgM high, and this is IgM high. So you can use just uh, diagrammatic representation of these three different types of mature B cells. Now, if we see the signaling through different antigens, then following maturation, the B cells they migrate to lymphoid follicles where there are two possibilities. One is that the B cell can be activated after antigenic interaction. So if the antigen is there, B cell will interact with it and then it will get activated. The other possibility is that the B cell recirculates through the blood and lymphatic systems and back to the lymphoid follicles. So it will go into the circulation and then it will come again to the lymphoid follicles. Now we have T dependent antigens, which are TD antigens. They bind to the Ig receptor. Okay, so here you can see these are the antigens, they bind to the Ig receptor. Now antigen is processed and presented to T helper cell. So here you can see the antigen has come here, pink antigen, and it is presented to the T helper cell with the help of the M.Sc molecules. T cell bind to the M.Sc peptide antigen and then delivering activating signals to the B cells via CD40 ligand. This is the CD40 ligand which is present on the T cell and CD40 which is on the B cell. So here you can see the CD40 on the B cell. So this is basically a activating signal. Now T cells also secrete activating cytokines IL-2 and IL-4 which are recognized by the receptor on the B cell surface. So you can see here these are the cytokines which are secreted by T cells and they are recognized by the receptors which are present on the B cells. Cytokines deliver differentiation, proliferation, and survival signals to B cells. So once these cytokines they bind here, they deliver the differentiation signal. So cell will start differentiating, right? Proliferation signal and survival signal to the B cells. Following T dependent stimulation, B cells may differentiate into plasma cells in the primary focus or early or low affinity memory cells, or may enter the follicles to participate in the germinal center reaction. Okay, so this can happen. Either they will form the primary cells at the primary focus, they can form the early low affinity memory cells, or they may enter the follicles to participate in the germinal center action. So what is the germinal center? We will see later here. Now then we have the T independent, and in T independent we have type 1 and type 2 antigens. So in case of type 1 antigens, they bind to B cells through both Ig and innate immune receptors. So you can see here this is the antigen. It is not only binding to the BCR, but also it is binding to the innate receptor which is TLR, which is toll-like receptor here. So example is the lipopolysaccharide from gram-negative organisms. They bind to B cells via both membrane-bound immunoglobulin and TLR4 signaling from both the receptors. So this cell, B cell will receive the signals from both the receptors, from the BCR as well as from the innate TLR receptors. Then the type 2, which is the T independent antigens, they are frequently bound by C3D complement components and cross-link both MIG and CD21 receptors on B cells. So here you can see this is the C3D, which is the complement, okay, component, and it is cross-linking with MIG and CD21 receptor on B cell. So cross-linking of between 12 and 16 immunoglobulin receptors by TI2 and antigens has been shown to be sufficient to deliver an activating signals. So once it is cross-linked by 12 and 16, between 12 to 16 Ig receptors by these type 2 antigens, then it is sufficient to deliver an activating signal and the cell will be activated. Okay. Now let's see, how the B cell antigen interaction takes place. So antigens after entering the body, it become concentrated in the peripheral lymphoid organs. Correct. Now if the blood-borne antigen is there, then it is filtered by the spleen, whereas in tissue spaces it is filtered by the regional lymph nodes. Now some low molecular weight antigens, which are their molecular weight is less than around 70 kilodalton, they enter the lymph node via a leaky network of conduits that are sampled by the follicular B cells. So follicular B cells, they sample these low molecular weight antigens, they simply leak into the conduits network. While high molecular weight antigens, which are more than 70 kilodalton, they are taken up by the Fc or complement receptors on the subcapsular sinus macrophages or by similar receptors on B cells, dendritic cells, and circulating macrophages, and subsequently they are passed on to the B cells, right? When the BCR recognizes its cognate antigen, okay, so BCR recognizes its cognate antigen, BCR's cluster, they representing the earliest phase of B cell activation. So BCR clustering represents the earliest phase of the B cell activation. So here you can see the cells, this is the B cell and it has the BCR, this is the antigen presenting cell, they are binding and then they are clustering. Okay, this is the earliest. Receptor clustering leads to the formation of an immunological synapse. This is the immunological synapse you can see here, where we have the peripheral and the central synapse. This is P stands for peripheral and C stands for central synapse between the B cell and APC. Now, if we see the B cell antigen interaction, then antigen engagement by the BCR induces a phosphorylation of the tyrosine residue in the ITAMs of Ig alpha and Ig beta by SRC family kinases. So here you can see these are the ITAMs, all right, and they phosphorylate. Okay. Now, this phosphorylation initiates a response leading to the formation of a cytoplasmic signal transducing complex, which we call it as a signalsome. So here this is the signalsome, and this signalsome is formed by BCR. Okay, so this is our BCR, right? Then it has lin and fin and BLK. Okay, so we have the lin, fin and BLK here, and then we have BTK, CD19, the adapter protein BLNK, BCAP, all right, and the enzyme PLC, gamma 2 and PI3 kinase. So that makes the complete signalsome. And there are multiple outcomes depending on the strength and duration of the antigen binding. So you can see here there are different outcomes here. There are different the transcription factors, and then they are transcribing the different cytokines. So I'm not giving much details here because we have already covered this in our signal transduction class. So therefore, if you want to go into the details of these pathways, you can just refer to that particular lecture. Further regulated by interactions between the BCR and other cell receptors, it includes CD21, CD40, IL4R, IL21R, and BAFFR. So these are some of the other cell receptors which are involved here. Now, the co-receptor CD21, it can bind C3D complement fragment associated with antigen and bringing the BCR into close contact with its co-receptors and enhancing antigen signaling. So basically this CD21, if you can see here, this is CD21 and then this CD21, it binds to, this is C3D, okay, complement fragment, and then bringing the BCR into close contact with its co-receptor, and then it increases the antigen signaling. Okay. Now, if we see the B cell antigen interaction, then B cells, they extract antigen from the antigen presenting cell membrane using active contractions of the actomyosin skeleton. So you can see here, this is the actin and this is the myosin, this is the clathrin. So this is these three, in the figure you can easily see that actin and myosin, they form the skeleton, actinomyosin, and here this is the BCR, and then this is the B cell, and this is the APC. Yellow one is the APC and green one is the B cell here. So how they are snatching, okay, so they are binding to the antigen and slowly they are taking away this antigen and they are forming a clathrin mediated endocytosis process, and finally the BCR mediated endocytosis transport antigen into vesicles. So here you can see they have formed a vesicle here where it is broken down into peptides. It will be broken down into peptides, and these peptides are subsequently loaded onto MHC class 2 molecules, and then they are returned on the B cell surface, and these MHC class 2 molecules and having the antigen, they interact with the T helper cells, and then T helper cells will provide another set of cytokine signals for further antigen, for their further survival and differentiation. Now, if we see the signaling through the different antigens, then T cell, they interact with their cognate B cells by binding to the processed antigen with the T cell receptor, as well as by interactions between the T cell CD28 with B cell CD80 and CD86, and between T cell CD40L and B cell CD40. So here you can see this is the B cell and this is the T cell. Now, the B cell and T cell, here you can see, these are the antigens here. This is the TCR, okay, and these are the receptors. So this is the CD28 and this is 40. All right. So here they are delivering the signals here. Clear? So if we see here, so MHC molecule, this is the MHC molecule loaded antigens here, okay, because and these antigens which are here on the MHC molecules, they are binding with the TCR here, okay, and ultimately there is a coordinate interaction, and then T cells activation differentiation takes place, and here the isotype switching or antibody production takes place here. Now these interactions, they facilitate the directional secretion of T cell cytokines that are necessary for full B cell activation, such as IL-4 and IL-21. Okay, so there is a directional secretion of T cell cytokines that are necessary for the full B cell activations, such as the IL-4 and IL-21, which are secreted by the T cells here. If we see the individual B cells, they may differentiate into plasma cells and early memory cells, or cells that enter the germinal center. Okay. Now, if we see the plasma cells, we have the naive B cells which bear cell surface IgM, but they do not secrete antibodies. So this is the naive B cell. Once antigen binds to them, it becomes the plasma blasts. Okay, they are differentiated to B cells that have begun to secrete antibodies. They have started secreting the antibodies. So you can see here the antibodies are secreted here, but they have not yet lost the capacity to proliferate and still bear the cell surface BCRs. So you can see some BCRs are there on the cell surface. They can still proliferate and they can also secrete. So this particular intermediate stage is known as plasma blast, and the next stage comes is the plasma cell, where the differentiated B cells can no longer divide, bear little to no cell surface. So you can see here the BCR is absent, and there is no proliferation, and immunoglobulin are rapidly surface immunoglobulin and rapidly secrete large number of antibody molecules. So you can see, as compared to the plasma blast, these plasma cells, they secrete very high number of antibody molecules, and there is no expression of membrane bound BCR, no expression. Okay. Now these plasma blasts and plasma cells, they are also referred to as antibody forming cells or AFCs. Okay. Now germinal center, you have heard this name so many times during our today's class. So what is it basically? If we see following T dependent stimulation, we are talking about the first kind of stimulation, which is T dependent stimulation. Some B cells enter the follicles and divide rapidly. The B cells, together with associated T cells and follicular dendritic cells, they form the germinal centers. So here we have the B cells, plus we have T cells, plus we have follicular dendritic cells, and they form a special centers which we call it as a germinal center. So here you can see germinal center how it looks like. So germinal centers have dark zones. So here you can see the dark zone, which is the dark blue color, where the B cells divide rapidly and undergo somatic hypermutation. And then we have the light zones. So here you can see this is the light zone, where B cells interact with T and follicular dendritic cells, and B cells bearing high affinity and mutated receptors are selected. So basically majority of the activity is happening here, while the preparation part is happening here in the dark zone, where we see proliferation and somatic hypermutation. Now, B cells in the dark zones, they are referred to as centroblasts. So we call them as a centroblast, while those in the light zones are referred to as centrocytes. So B cells here are called centrocytes, and in dark zone they are called centroblasts. Now, movement between the two zones, it is orchestrated by modifications in the cell surface chemokine receptors. So as the cell surface chemokine receptors change, so chemokines, they direct the movement from dark zone to light zone or light zone to dark zone. Now, germinal center B cells, they are prone to apoptosis. Survival requires productive interactions with T cells as well as with the follicular dendritic cells. So as long as the T cells are coming, sorry, uh, as long as the B cells, they are interacting with the T cells and follicular dendritic cells, they will be survived. Rest of the cells, they will not survive. Why? Because as long as the antigen is there, the interaction will be there, and the interaction will be there, we need the B cells to survive. And if the antigen is absent, then no need to interact with the T cells or the follicular dendritic cells. Therefore we don't need the cells, and therefore through apoptosis they are eliminated. Okay. Now, somatic hypermutation, this is very important event, and this is I think very unique event in B cells. So particularly if we see in germinal center, this somatic hypermutation, it introduces point mutations into variable region of Ig genes or the antibody genes, right? These mutations in the variable genes, they are initiated by an enzyme called activation induced cytidine deaminase, which is AID. This enzyme is important for somatic hypermutation, which is activation induced cytidine deaminase or AID, which is expressed only by germinal center B cells, right? So you can see, why germinal centers are only, place where we see the somatic hypermutation, because that is the place where AID is expressed, and this is expressed only by the germinal center B cells. These mutations, they can increase or decrease the affinity of antibodies for the specific antigen. So basically, they optimize the affinity of the BCR towards the antigen. So B cells producing higher affinity antibodies, they are selected to survive because they have to capture more antigens and proliferate, which is known as affinity maturation. So this particular event is known as affinity maturation, where B cells, they produce higher affinity antibodies and they are selected to survive and proliferate. It is critical for generating highly specific antibodies and enhancing immune defense against evolving pathogens. And it contributes to long term immunity after vaccination or infection. You know, this is important or critical for generating highly specific antibodies, for enhancing immune defense against evolving pathogens, and contributing to long term immunity after vaccination or infection. The activity of AID, which is expressed only in B cells, it requires access to the cytidine side chain of a single stranded DNA molecule. This is the prerequisite for the activity of the AID. It requires the access to the cytidine side chain of the single stranded DNA molecule. So AID initiates a nucleophilic attack on the exposed cytosine ring. So here you can see this is the cytidine side chain, and here the AID, it attacks there. So single stranded DNA is attacked by the AID, all right, and it is resolved by the deamination of the cytidine to form uridine. So here this is the AID transition state, and then finally there is a regeneration of AID and formation of uridine, correct? So AID initiates DNA lesions whose repair leads to somatic hypermutation, and we through the class switch recombination or gene conversion. So basically it is causing what, a single stranded DNA nucleophilic attack, and then during the repair there are chances that there would be the point mutations, and that produces the variability or the somatic hypermutations. So class switch recombination, it results in the replacement of an antibody heavy chain constant region with a different constant region. So you remember we have the 5 different types of heavy chains: MADG, IgM, right, IgA, IgG, IgD and IgE. So what is this class switching recombination is doing? It is basically replacing the antibody heavy chain constant region with a different constant region, but it is not touching the variable region. So that means the specificity towards a particular antigen, it remains same, no change, while the constant region changes, so the class changes, and therefore we call it as a uh, class switch recombination, because one class is switched to another class. Now, somatic hypermutation, it occurs only in the GCs and only after the T-dependent antigen stimulation. That is the rule here for somatic hypermutation. It occurs first of all only in the germinal center, correct, and only after the T-dependent antigen stimulation, correct. Now, class switching recombination may occur elsewhere and in the presence or absence of help from T cells. So, this is the difference between the somatic hypermutation and class switching recombination. While somatic hypermutation, it occurs exclusively in germinal centers and only after the T-dependent antigen stimulation, while class switch recombination may occur elsewhere, right, and in the presence or absence of help from the T cells. Okay, now look at the memory cells. So IgM bearing memory B cells are generated early in the primary response, prior to the onset of the somatic hypermutation, prior to the onset of the somatic hypermutation, and secrete non-mutated antibodies of lower affinity. So these are the memory cells which actually originally, they remain original in the chain, because they are formed prior to the onset of the somatic hypermutation, and they early in the primary response. As we go to the towards the late response, then there are chances then somatic hypermutation will take place, and therefore the memory cells will not be, that this memory cells will not form, and they secrete non-mutated antibodies of lower affinity. So that means affinity maturation has not achieved yet. While IgG bearing memory cells, they are generated later in the primary response. They have mutated and selected receptors, and they yield recall response that deliver high affinity antibodies. Okay, so here is the difference between the IgM bearing memory B cells and IgG bearing memory B cells. So IgM early, IgG late. IgM, there is prior to the onset of hypermutation, while they are secreted after the hypermutation, and then IgM bearing memory B cells, they secrete non-mutated antibodies to lower affinity, while IgG bearing memory cells, they secrete high affinity antibodies. Now we see the T independent responses. So T independent responses are generated by B1 and marginal zone B cells. They give rise to relatively low affinity and primarily IgM class of antibodies. Most T independent antigens are carbohydrate in nature, and T independent type 1 antigens, they interact with B cells via both the BCR and innate immune receptors, whereas T independent type 2 receptors, they are highly polymerized antigens that do not bind to the innate immune receptors. That is the major difference between the T independent type 1 antigens and T independent type 2 antigens. Okay, now both types of T independent responses, they are enhanced by interactions with other cell types, including T cells, macrophages and monocytes and neutrophils. All right, so these cells, they increase or enhance the interaction, right? Now if we see the mature B cell subsets, then we have the conventional B2 B cells, then we have the B1A and B1B B cells and marginal zone B cells here. Now this slide is slightly what we have already discussed, but it gives some slight more information. Okay, so if you can see here, the conventional B2 cells, the major sites are secondary lymphoid organs, while here it is pleural and peritoneal cavity and spleen and marginal zones, they are in the marginal zones of spleen. Now, if we see the variable region diversity, these are highly diverse, these are more restricted in diversity and moderate diversity. And rapidity of antibody response is slow. This is rapid and rapid in case of B1 and marginal zone. Surface IgD is high levels of IgD, whereas low levels of IgD, and here also low levels of IgD. Somatic hypermutation is present in conventional B2B cells and marginal zone, and absent in B1B B cells. And requirement for help, so here in case of conventional B2B cells, it is provided by T cells, while B1A B cells, they are the T cells help not required, while T and other cells can enhance response, although help is not required. Whereas in case of marginal zone B cells, the T cell help not required, while T cells, dendritic cells and neutrophils can enhance the response. So help is needed here in the conventional B2B cells. They participate in germinal center reaction, definitely yes in conventional B cells, while no in case of B1A and B1B B cells, while it may be possible in case of marginal zone B cells, although with slower kinetics and follicular B cells. Isotypes produced are all isotypes, all five types are produced by conventional B2B cells, while in case of B1A and B1B cells, predominantly they are IgM, and marginal zone B cells, these are also predominantly IgM. If we see immunological memory, yes, in case of conventional B2B cells, very little in case of B1A and B1B B cells, while source of IgM producing memory cells are there or have been found in case of humans in marginal zone B cells. Okay. Now, if we see the mature B cell subsets, then we have the regulation. So, effector B cells, they positive regulate immune responses through provisions of IL-6, IFN gamma, and GM-CSF. So, here you can see this is IL-6 B cells, IL-6 here, and IFN gamma. They are the effector B cells here. They do the macrophage activation by IFN gamma and GM-CSF, while IL-6, it produces the plasma cell differentiation. All right, and then IL-6 here also it causes the Th17 differentiation. Whereas, the regulatory B cells, they negatively regulate immune responses through provisions of IL-10, IL-35 and TGF beta. So this is the negative regulation here. IL-10 and IL-35. You can see here inhibition of Th1 and Th17, IL-10, IL-35 and TGF beta for dendritic cell inhibition, and IL-10, IL-35 and TGF beta, they increase the regulatory T cell differentiation. So this is the negative regulation, and here you see using IL-6 and IFN gamma and GM-CSF, it is the positive regulation. They are secreted by the B cells. Okay, so here this is about the B cell development pathway, and let us close the discussion of today's class, and let us see what we learned today. We discussed about the B cell development niches, B cell development pathways, types of B cells, and B cell maturation. All right, in case if you have any questions or you have some doubt, you can either go through this presentation again, or you can just refer to your textbook. Okay, and just take care of yourself, and thank you very much.